Welcome to my channel XY Biology Academy. Imagine two bacteria that are actually the same species. One appears as a smooth white colony on a petri plate. The same bacterium produces cloudy broth in a test tube. And on a gelatin tube, it even liquefies the medium. How is that possible? Did the bacterium change? Answer is no. The bacterium is the same. Only the environment is different. And just like humans behave differently at home, in school, or on a playground, microorganisms also behave differently depending on the medium in which they grow. And the principle behind these differences is that every microorganism has its own unique genetic makeup. Because of these genetic differences, microorganisms grow differently, produce different pigments, secrete different enzymes, and spread differently on culture media. When microorganisms are grown on different laboratory media, they exhibit differences in their visible appearance. These visible differences are called cultural characteristics, and microbiologists use them as one of the first clues for identifying unknown microorganisms. Although cultural characteristics alone cannot confirm the identity of a microorganism, they significantly narrow down the possibilities before biochemical or molecular tests are performed. These characteristics have been systematically described in Bergey's Manual of Systematic Bacteriology, the world's most widely used reference for bacterial classification and identification. In this lecture, we'll learn every cultural characteristic observed on nutrient agar slants, nutrient agar plates, nutrient broth, and nutrient gelatin, and, most importantly, understand why these characteristics differ from one medium to another. First, we describe cultural characteristics on nutrient agar slant. A nutrient agar slant contains solid agar inside a test tube with a slanted surface. When bacteria grow on an agar slant, we observe 5 important characteristics. Number 1 is abundance of growth. The word abundance literally means how much of something is present. Here, it simply means the amount of bacterial growth. Growth may be no growth, slight growth, moderate growth, or heavy or abundant growth. The amount of growth depends on factors such as nutrient availability, incubation time, temperature, oxygen, and the growth rate of the microorganism. Fast-growing bacteria usually produce abundant growth, while slow-growing organisms may produce only slight growth. Number 2 is pigmentation. Pigmentation refers to the color produced by microorganisms. The word pigment simply means color. Some bacteria synthesize pigments inside their cells. These are called intracellular pigments. Others release colored substances into the surrounding medium. These are known as extracellular pigments. For example, some species produce yellow, red, green, blue, or orange pigments. However, most bacteria are non-chromogenic, meaning they do not produce pigments and therefore appear white, cream, or gray. Pigments may help microorganisms protect themselves against sunlight, oxidation, or environmental stress. Number 3 is optical characteristics. The word optical relates to light. Optical characteristics describe how much light passes through bacterial growth. There are 3 possibilities. First is opaque. It means no light passes through. The growth completely blocks light. Second is translucent, which means only some light passes through. Objects behind the colony appear blurry. The third possibility is transparent, which means almost all light passes through. The growth appears nearly clear. These differences depend on cell density and colony structure. Number 4. Form. Form simply means the overall appearance or pattern of bacterial growth along the inoculation line. Let's understand each type one by one. Filiform. The word filiform comes from the Latin word meaning thread-like. Growth appears as a smooth continuous line. The edges remain even and regular. Acuminate. The word acuminate comes from a word meaning spiny or prickly. Growth remains continuous but has rough, irregular edges. Beaded. The word beaded literally means resembling beads on a necklace. Instead of one continuous line, separate colonies appear along the streak. Effuse. Effuse means spreading outward. Growth becomes thin and spreads widely over the agar surface. Arborescent. Arborescent comes from the Latin word arbor, meaning tree. The growth forms branches resembling a tree. Rhizoid. Rhizoid comes from the Greek word rhiza, meaning root. Growth develops root-like branching patterns extending outward. Number 5. Consistency. Consistency describes how bacterial growth feels or appears when touched with an inoculating loop. Dry colonies appear rough and lack moisture. Buttery colonies are smooth, moist, soft, and resemble butter in texture. Mucoid colonies are slimy, sticky, shiny, and usually indicate the production of a polysaccharide capsule. Capsule-producing bacteria often show mucoid growth. Now we will see cultural characteristics on nutrient agar plate. Unlike an agar slant, the agar plate provides a large flat surface where isolated colonies can develop. Each colony usually originates from a single bacterial cell or a small cluster of cells. Because colonies are isolated, we can study many additional characteristics such as size. The word size simply refers to how large each colony becomes. Colonies may be pinpoint, small, medium, or large. Colony size depends on the growth rate of the microorganism and incubation period. Pigmentation. Exactly as discussed for agar slants, colony color may result from intracellular or extracellular pigments. Colony form. Form refers to the overall shape of the colony. Circular colonies have smooth round edges. Irregular colonies have uneven outlines. Rhizoid colonies resemble spreading roots. Margin. Margin literally means the edge or boundary of the colony. Entire margins are smooth. Lobate margins have deep rounded projections. Undulate margins are gently wavy. Serrate margins look like the teeth of a saw. Filamentous margins contain thin thread-like extensions. Remember that form describes the entire colony shape, whereas margin describes only the outer edge. This distinction is commonly tested in examinations. Elevation. Elevation tells us how high the colony rises above the agar surface. Flat colonies have almost no elevation. Raised colonies are slightly elevated. Convex colonies form a dome. Umbonate colonies possess a raised central bump. Now think that if two colonies have exactly the same shape but different margins, are they considered identical? Answer is no, because margin is an independent identifying characteristic. Now observe cultural characteristics on nutrient broth. Nutrient broth is a liquid medium. Since there is no solid surface, bacteria cannot form colonies. Instead, we observe how they distribute themselves throughout the liquid, and following characteristics can be observed. Uniform fine turbidity. The word turbidity means cloudiness. Uniform fine turbidity indicates bacteria are evenly dispersed throughout the broth. Flocculent. Flocculent comes from a word meaning flakes of wool. Cells aggregate into floating clumps throughout the broth. Pellicle. Pellicle literally means a thin skin or membrane. A thick bacterial layer develops on the liquid surface. This often occurs in organisms requiring abundant oxygen. Sediment. Sediment refers to material settling at the bottom. Heavy bacterial cells accumulate at the base of the tube. Sediment may appear granular, flaky, or flocculent. Why do some bacteria grow only on the surface while others remain throughout the broth? Answer is because different bacteria have different oxygen requirements. Aerobic bacteria usually concentrate near the surface where oxygen is highest, while other organisms distribute differently depending on their oxygen needs. At the end we will see cultural characteristics of microbes in nutrient gelatin. Nutrient gelatin is unique because it helps detect whether bacteria produce the enzyme gelatinase. Gelatin normally remains solid. If bacteria produce gelatinase, they digest gelatin, causing the medium to liquefy. The pattern of liquefaction becomes an important identifying characteristic. So here you can observe different characteristics like crateriform, looks like a shallow crater. Only the upper surface becomes saucer-shaped. Napiform. The word napiform means turnip-shaped. Liquefaction forms a bulb near the top. Infundibuliform. This word means funnel-shaped. Liquefaction gradually widens downward like a funnel. Saccate. Saccate means sac-like. Liquefaction forms an elongated tubular cavity. Stratiform. Stratiform means layer-like. The upper portion of the gelatin becomes completely liquefied. Let's quickly revise. Agar slants help us observe abundance of growth, pigmentation, optical characteristics, form, and consistency. Agar plates allow us to study colony size, pigmentation, form, margin, and elevation. Nutrient broth reveals how bacteria distribute themselves in liquid media. Nutrient gelatin demonstrates gelatinase activity through different liquefaction patterns. Although the microorganism remains the same, different culture media reveal different characteristics because each medium provides a different physical environment and allows different microbial properties to become visible. That is why microbiologists never rely on a single culture medium for identification. Instead, they combine observations from multiple media to build a much more accurate picture of the microorganism before proceeding to biochemical and molecular confirmation.